This paper looks at how an oral infection of COVID can lead to a progressive sequence of events that causes a nasal infection of COVID and an olfactory bulb infection of COVID. Using a Syrian hamster model, they explored whether an oral infection of COVID can lead to a nasal infection. And on day 7 post infection, COVID presence was confirmed in the lateral area, but not the nasal septum of these positive mice, where the virus was found from the olfactory nerve bundles to the olfactory bulb, suggesting that the nasal cavity is a route for COVID brain infection. So, in this paper, they demonstrated that transoral COVID infection can spread from the nasal cavity to the central nervous system and the possibility of central olfactory dysfunction due to COVID infection. So, backing up a little bit, that olfactory dysfunction, what is that? Well, that's going to be your loss of smell and subsequently your loss of taste, since taste and smell are related. A study found that in Syrian hamsters, the olfactory epithelium is extensively disrupted in the first few days after infection, and that the loss of microvilli becomes significant on days 2 to 7. So we're starting to see impacts within 48 hours. So, what is the olfactory epithelium? It's sort of the back cavity of the nose, where just a thin area of bone separates this cavity from the brain, and there are nerve roots that sort of connect between the two. Where the olfactory bulb sits on the brain side, and the olfactory epithelium is on the nasal cavity side. And interestingly, like we've seen in other nasal studies, the virus mainly infects the microvilli cells. And after this damage occurs to the olfactory epithelium, regeneration begins approximately on day 8 and is almost restored by day 21. But this regeneration is insufficient in the lateral and dorsal regions, meaning that the regenerative process can vary depending on the location in the nasal cavity. To cause infection in these Syrian hamsters, they did 50 microliters of virus solution diluted in saline and administered into the mouth, while the control group was given 50 microliters of saline only. Following the infection process and the incubation period, they did histology after euthanasia of the animal. So the first interesting finding is see how everything is nice and intact. Over here is the COVID infected hamsters, and you can see that there's a lot of disruption. There are little pieces sort of cut away. It's very thin. That's what it means by the damage to the nasal epithelium. And all of the pinkish red in this image, that is COVID that is present in those areas. Where we largely only see COVID in the lateral areas, so the side areas, and interestingly, COVID entering the host animal via the oral route not only infected the lower respiratory tract, but also those nasal tissues, which means that inhaling droplets from conversations during eating and drinking, or by eating something that had been touched with COVID contaminated hands, the virus is likely to adhere to the oral cavity and enter the body. Therefore, individuals should thoroughly wash their hands before eating, be careful of splashing droplets during conversation while eating, and they should avoid sharing meals with others to prevent infection. And recall that if you sit side by side with someone that you're sharing a meal with, you're much more likely to not get that fomite contact while eating the meal. But when you sit across from one another, you can have those issues. So that's a little risk mitigation strategy we saw in a prior paper. So this study shows that ultimately, yes, you can get a COVID infection by ingestion, by fomite contact, consumption of COVID in your mouth.